Good morning, everyone. I hope you're doing well in this crisis. I hope that everything will go well. In the meantime, let's benefit from our time. I was thinking of doing this video to try to work together on lesson 3, which is digestion. So, what is digestion? Digestion is a set of mechanical and chemical molecular simplifications that are taking place in of food into nutrients. So, from macromolecules into micromolecules. To understand in vivo digestion, or the digestion that takes place in our body, we need to understand, first of all, the in vitro digestion, which is digestion or artificial digestion that takes place in test tubes. For this reason, we're going to look at different experiments that were done to be able to understand the in vitro digestion, and from there move on to the in vivo digestion. So the first experiment that we're going to see is on page 18, document B. This experiment we're seeing the in vitro digestion of cooked starch. So let's look at this together. The question is to describe the experiment in document B on page 18. What do I mean by describe? When we say describe, it means we are going to say just the steps that were done to do this experiment. So in the back of my mind, I need to put this. I need to remember that when I'm describing, it is as if I am simply doing the experiment and I'm putting the steps. It's like baking a cake. Same logic over here. So you just have the steps or you're writing the steps or the procedure that was done. So let's look at the experiment together. So you have the schematic interpretation and you have the setup of the digestion of cooked starch in document A, and the schematic interpretation, document B. We can see two test tubes that were put in or placed in a water bath. These test tubes, they contain both of them cooked starch, and in one of the test tubes, which is tube A, we have a small amount of fresh saliva. The water bath has a temperature of 37 degrees Celsius. So, given all this, what are we going to, or how are we going to describe? We're going to say, we put in two test tubes, A and B, cooked starch. We added, and you can see over here with me, that there is the plus sign, which means that you added. We added a small amount of fresh saliva in tube A. We placed both tubes A and B in a water bath at 37 degrees Celsius. And if we continue reading under the documents A and B, we can see that these two tubes were placed for 15 minutes. So, if I want to write my description, as I just said this, put cooked starch in both tubes A and B, add a small amount of fresh saliva in tube A, place the tubes in a water bath, temperature 37 degrees Celsius, for a duration of 15 minutes. Please, everyone, just remember, when you're performing the experiment, you cannot place the two tubes inside the water bath, then you put substances or things inside, because you aren't able to perform the experiment like this. So these are the steps that were done to be able to do the experiment. Moving on. Second part says, interpret the results in document C on page 19. Let's look at the results together. So we can see that we have two tests that were performed. First test is the identification test, which is iodine test, and the second one is the Fehling test. Now, if you noticed, this is a bit tricky. Why is it tricky? Because the colors are the same for both of the tests. But now you need to take this further. What do I mean by taking it further to the next step? Let's start with the first test, which is the iodine test. Usually, iodine gives a blue color in the presence of cooked starch. Excellent. And if we don't have cooked starch, it gives an orange brownish color. So, based on what we already know, let's try to interpret over here. In the first test, which is the iodine test, both tubes A and B gave a positive result at time 0. All right? Then, after 15 minutes, tube A gave a negative result, which is the orange brown color, and tube B gave a positive result for iodine solution. What do I mean over here? Why did tube B remain the same, or why did it stay blue in color and the tube A changed its color? So simply because digestion took place. No more starch is there. So when we do not have starch, what does this mean? That the absence of starch means the digestion of starch took place. All right? As for the second experiment, which is the Fehling solution, both tubes A and B at time 0 gave a negative result, true. In tube A, after 15 minutes, a brick red precipitate appears, indicating the presence of a reducing sugar, true. So, whereas in tube B, do we have a reducing sugar? No, because the result stayed negative. What does this indicate? Both of these tests, they were done to be able to show us that in tube A, digestion took place, whereas in tube B, no digestion took place. So, if I want to write my interpretation, what am I supposed to say? Let's look at this together. So, at time 0 minutes, we perform the iodine test on each tube. The iodine test gives a dark blue color in each tube, indicating the presence of starch. The Fehling test gives a blue color, indicating the absence of a reducing sugar. 15 minutes later, the dark blue color changes to a brown color, indicating the absence of starch in tube A, whereas the dark blue color remains in tube B. As for the Fehling test, a brick red precipitate appears, indicating the presence of a reducing sugar resulting from the digestion of starch in tube A, whereas in tube B, the color remains the same. Saliva contains the enzyme salivary amylase that hydrolyzes cooked starch into maltose in a neutral medium at a temperature of 37 degrees Celsius for a duration of 15 minutes. So, in conclusion, with all of this, we can conclude that saliva digested the starch, or the presence of salivary amylase that is found in saliva digested starch, and it produced maltose. We also have another enzyme that works on starch, but over here this enzyme is found, or it's secreted by the pancreas. This is called pancreatic amylase, which hydrolyzes both cooked and raw starch into maltose, and over here the pH is supposed to be basic and not neutral, right? This is an application exercise that I'm going to post on the agenda, the daily agenda, that you can try to work on. And then later on, I'm going to post the correction of this. So, and you can see that the correction is also found over here. We're going to look now at the in vitro digestion of proteins. We have to note together, and as you've seen this last year, pepsin is an enzyme that hydrolyzes proteins into peptides. How does it hydrolyze, or in what condition does it hydrolyze proteins into peptides? It's supposed to be, we're supposed to have acidic medium, temperature which is 37 degrees Celsius, and the duration is supposed to be 1 hour. So let's look at the document A on page 22, and also the question over here is to describe the experiment. So let's describe it together. Remember, put, add, place, duration, and the temperature. So we put coagulated egg white in a test tube. We add pancreatic extract. We place the tube in a water bath for a duration of 1 hour at 37 degrees Celsius. Do we need to say what happened over here with the result? Is the question about the result? No, the question is to describe the experiment. Put coagulated egg white in a test tube, add pancreatic amylase, pancreatic extract over here, and place the test tube in a water bath at a temperature of 37 degrees Celsius for a duration of 1 hour. Pancreatic juice contains enzyme which is trypsin. The trypsin hydrolyzes proteins in a basic medium at a temperature of 37 degrees Celsius, and it produces amino acids. All right. Also, I'm going to ask you to do exercise 10 on page 31 if you don't have your books. I'm going to screenshot this and I'll post it on the agenda. And the results of the experiment or the result of the exercise. Even exercise 11. All right. In vitro digestion of lipids. Let's all look at the experiment that was done on page 22, document C. Can we describe this experiment together? Same logic. We put oil in a test tube, we add water to it and bile. We shake the test tube well and we add pancreatic extract. We place the test tube in a water bath at 37 degrees Celsius. And if you can go further, we are able to see that this needs approximately 3 hours. All right, but if it's not there, if we don't have the duration, you do not need to add the duration. So the word pour over here is better than what I said before, which is put, because you're pouring a liquid. So you pour oil and water in a test tube, you add bile and you shake the tube well. Then you add pancreatic extract, you place the tube in a water bath at 37 degrees Celsius. We need to pay attention that pancreatic juice contains the enzyme lipase, so the extract has lipase, and lipase works on hydrolyzing lipids in the presence of bile to fatty acids and glycerol. What's the function of bile? As you've seen this last year with Mr. Elias, it's a greenish liquid secreted by liver, which is stored in the gallbladder. What does it do? It provides a basic medium. It emulsifies lipids, that is, it breaks it down and makes the lipids into small droplets so that it would be easier to digest. What do you mean by emulsification? The word emulsification, it means that it removes the lipids from each other or segregate them. All right. Then emulsification of lipids, is it a chemical digestion or a mechanical digestion? No, it's a mechanical digestion, because over here there is no chemical reaction that is going to take place, or there is no change in the composition of the lipids. The lipids are going to stay as is. They're going to stay the same. All right. Based on all of this, based on in vitro digestion of the lipids, results of an experiment that was done, of this experiment that we just described were done, and we can see the results in the graph below. The graph is taken from the book, page 22, document D. Let's look at the graph together. We can see 2 lines on the graph. First of all, it's very important to look at the x-axis and the y-axis. And remember, What's the dependent variable over here and what's the independent variable over here? What is happening over here? What is changing with respect to what? So you have on the y-axis the level of measured substance. Why did we write the level of measured substance? Because we have two substances that were measured. First one is the oil and the second one is the fatty acid. So the level of the measured substance, how is it changing with respect to time? So as the time is moving on, we're seeing the change in both of the fatty acids and oil. So if I want to analyze this over here, we have a graph, and this needs a comparative analysis. Why do we need a comparative analysis in this case? All right, we need a comparative analysis in this case because we have two things that are, or two substances that are changing with respect to time, but they are also changing, right, with respect to each other. So if you noticed, at time 0, the amount of fatty acids, it was 0. And at time 0, the amount of oil was 100%. As the time increased, right, the amount of oil decreased sharply? No. Steadily? No. Slowly? Yes. So it decreased slowly to reach approximately 0% at time 180 minutes, which is 3 hours. As for the fatty acid, it increased from time 0 till time 180, also slowly, and reached 100% at time 180 minutes. Now, shall I go further with this? No, because over here I am simply analyzing the graph. So the graph that is showing us over here is a graph of the variation in the level of oil and fatty acid in percentage as a function of time in minutes. So at time 0 minutes, the level of oil is 100%, whereas the fatty acid is 0%. So I need to compare over here. This is why I have the word whereas. The level of oil decreases to 0%, whereas that of fatty acid increases to gradually or slowly to 100% at time t equals 180 minutes. Now, what conclusion can I draw out? Let's think about this together. Okay, so oil is digested to fatty acids. In the presence of what? In the presence of pancreatic extract or the enzyme lipase. Right, I want you also to try to work on exercise page 30, number 9. And also if you don't have your books, I'm going to send you the screenshot of this on the agenda. And later on, after a few days, I'll be posting the answers for exercises 9, 10, and 11. All right, thank you everyone, and I hope that this video was up to your expectation.